Hi everyone. I go by the name Ani. I'm a postdoc with Eugene Shaknovich, and a disclaimer that I'm pretty new to this, the whole field of microbial evolution and ecology. So it has been so much fun and learning experience for me to this amazing community here. And the work I'm going to present today is very much work in progress. So I would very much appreciate if you have any sort of feedback. So to give a little bit of my research background, the kind of problem I have been thinking of for last several years now is this problem of biomolecular condensates and phase separation. So this is some kind of a generic way that cells sort of organize material in space. So as you can see, it is really a multi-scale problem that starting from biopolymers, you can go way all the way up to the cellular scale. And then there are different kinds of questions you can ask using different kinds of biophysical simulation techniques, right? So on the right-hand side, we have this molecular scale where we can use something like coarse grain molecular dynamics or bioinformatics. And on the middle scale, we have this so-called mesoscale, where we have this biomolecular condensates, which is really like a network of molecules which are doing interesting stuff in signaling, metabolism, and so on and so forth. And then we have on the extreme left, we have the cellular scale, where we can employ techniques like dynamical systems theory, including ODs and PDs, depending on the context that whether space is important or not. By training, I'm a systems biologist, so I'm generally very much interested in any sort of complex adaptive systems, which essentially includes this complex interplay between kinetics, equilibrium, and feedbacks. And I'm recently, as I mentioned, started thinking about this spatial organization as well. So that's why I was curious to come to this meeting to sort of get a perspective of people's thinking, although most of the people are talking about slightly different scale, but I think the underlying physical rules could be same. Um, so the story that I'm going to share today is related to like a microbial resistance. So we are thinking this problem in the language of microbial evolution, and I'm sure most of you in this room are familiar with this problem of AMR, which is a global health crisis. So in this cartoon, what I'm trying to show here, that you put some kind of a chemical stress in the form of antibiotic, that will alter the physiology of the organism. But eventually, the organism will evolve out of the situation, right? So that will result into some kind of a partial or full rescue of the physiology. And the physiology could be metabolic state, for example. So historically speaking, Shakhnovich lab is interested in protein evolution. So the kind of problem that they're interested in, that what drives the structural stability of a protein molecule, for example, and what happens if you trigger point mutation to the individual structure, and what kind of functional consequence those mutations might eventually have. So on the left-hand side, we have this very finer molecular scale where we are talking about the structure of individual enzyme. And on the right-hand side, we have this growth kinetics of on a population level of some bacteria, for example. So this is this, like a very physicsy idea of genotype to phenotype mapping. And the question here is, what happens when you trigger a point mutation to the structure of individual enzyme, and how does it map back to the population level? Okay, so that's the kind of question that this group is historically interested in. And and of course, there are several different classes of antibiotics. One of the important classes is this, like blocking the metabolism. So, for example, folic acid is one of the essential metabolites that this bacteria need to synthesize in order to survive. And one strategy of devising antibiotics could be to block that synthesis of folic acid. So amongst several classes of mechanism, this antifolate is one of the strategy to design antibiotics. However, there is a problem, of course, that this TMP molecule, for example, is a classical case of antibiotics. But as I mentioned, that organisms evolve out of that stress, right? So in this particular paper, the experimental side of our group, what they did that they could successfully synthesize a small molecule which not only targets the wild type, but it also targets the mutant version of the protein, for example. And those mutations eventually lead to the escape of that stress. So by designing this kind of compound, we can think of this somewhat ambitious goal of evolution drug, meaning it not only blocks the current variant, but it can also block the escape variant to some extent. So this is sort of experimental demonstration that in the presence of TMP and this new compound CD153, you can see the rate curve that the bug is having a hard time to evolve out of that chemical stress, basically. Okay. In a follow-up paper, my colleague Saurav and others, they also showed that it's also possible to design this small molecule which can target multiple enzymes at the same time, not just one, but maybe many. And if you do that, then again, you have this advantage of constraining the evolvability of the pathogen. Okay, so that is sort of the motivation of this work. And of course, this is just I put this cartoon to put things in context that biology is, of course, a very multi-scale problem. So we have this hierarchical organization across different scale, all starting from molecule all the way up to the organism. And in this interesting multi-scale problem, going from molecular scale to the organism scale, there is this very interesting intermediate layer, which is this metabolic network in this case. So we can imagine that, on active site level, there are several point mutations that are feasible. But the question is, what kind of effect it will have on a network level? Because network might work as some kind of a regulator which can wash out some of the effect. Okay, so this network sort of puts this additional interesting regulatory layer, which we really need to understand if we want to come up with a very efficient protocol to design this therapeutic intervention. So that's sort of the goal of this project, that to fill in this gap between these two extreme scale and try to see what, by using some very simple physical models, what can we learn about this whole antibiotic resistance problem. So as I mentioned, there is some long-term and short-term goal. So some of the short-term goals would be, what are the dynamics and steady state of the pathway of interest, and some of the long-term goal would be, okay, if we encode space into this topology now, like can we get some extra advantage or disadvantage? So those are the kind of short-term and long-term goals of this kind of direction. And we are thinking the problem in some kind of a modular approach. So for example, let's say this is again a very cartoonish version of the whole metabolic network of the full organism, and we can decompose the full network into multiple small modules. So let's say we're interested in pathway 2. So the way to model that would be, there is an influx coming from pathway 1, and then there are 2 outflux going into pathway 3 and pathway 4. And we're interested in zooming into this pathway 2, basically, and we are going to model all the metabolic reactions that are part of pathway 2. Okay. So as I mentioned, we're interested in folate biosynthesis. So this is again like a cutout version of the cake database, and these are the like a different kind of reactions that are part of the folate biosynthesis. So this is sort of our current representation that we are working with. So we have 28 nodes or 28 metabolites, and we have 35 reactions, and we have 3 input points and 5 output points. Okay. And each of these 35 reactions has an enzyme. That means each of them has a Km and Vmax. So a word about the reaction kinetics here, that we have this reversible Michaelis-Menten representation for individual reaction. So for a reversible Michaelis-Menten, somewhat complicated looking form appears. But one interesting thing to realize, which has been a central discussion point for this meeting also, that equilibrium is important. So there is this so-called Halden relationship, which puts some constraint, and also it connects the K equilibrium with this kinetic parameter like Kcat and Vmax and so forth. But one interesting thing to note here, that within this constraint, there are several ways of choosing this Km and Vmax, and that will give some kind of evolutionary, like a buffering zone without violating this constraint. And I think that is something to keep in mind when we are designing this kind of system, because within this thermodynamic constraint, one can choose multiple values of these kinetic parameters without violating this careful interplay between kinetics and thermodynamics. So we can again take some spherical curve approximation and make some assumption to simplify the expression. So for example, we can assume that okay, the Km for the product and substrate is same, and the Vmax reverse is basically Vmax forward over K equilibrium given by this Haldane constraint. So this slide sort of shows you this kind of a very toy model, but it also highlights the fact that equilibrium and kinetics has different role in the overall dynamics of your system. So here what I'm showing that I have three different values of K equilibrium, keeping both Vmax and Km same. And you can see that the kind of reaction, the amount of product you have in a system is different. But now you can do the opposite. You can keep the K equilibrium fixed and change the rate parameters in your system. You don't change the relative proportion of the substrate and product, rather you change the rate at which it converts to those solution, right? So those are the kind of the interplay between the kinetics and equilibrium. Another interesting thing to notice here that for me, for example, before starting this project, I was mostly I used to think chemical kinetics in the language of mass action, but the purpose of this demonstration is to show that mass action has some kind of behavior which is not consistent for enzyme kinetics. So, for example, let's say we have this source, reaction, and sink type of framework. Doesn't matter how much stuff you put in your system, mass action will always sort of process that system to reach to a steady state. But definitely that's not the case for enzyme kinetics, because enzyme kinetics has some kind of a capacity, because you have this Vmax which puts some kind of upper constraint. So even though you have a reversible reaction, just because of this saturation of this enzyme core, you have this some kind of a accumulation of stuff in your system, which was not realized if you are just using simple mass action to model this reaction. So now getting into somewhat interesting regime of this type of model. So let's say we are considering a toy network here, right? So these are the bunch of reactions. Now we have this interesting loop in the middle, and let's say hypothetically speaking, we design two inhibitors which can inhibit R2 and R5 respectively. So these are, at the absence of those inhibitor, you have some steady state value of your system. Now, in the presence of this inhibitor, you get some kind of alteration. As you can see that all the curves are moving up and down. So to compute that, we came up with this parameter called divergence quantification, which basically takes the difference between two probability distribution you have. So let's say you have a wild type distribution P and you have some kind of a mutant distribution Q. So by this sort of KL divergence, we can compute what is the extent of perturbation in your network. Okay. And these are the different fitness function that is not very important for the context of this talk. But the important thing to notice here is that let's say we have only one inhibitor present in your reaction, which is I1. And remember, you have more divergence means more disruption. That means if you have more divergence, you are disrupting the physiology more to the organisms, which is a bad news for the organism. But interestingly, once you put I2 into your system, your divergence gets reduced somehow, which is a fairly non-trivial thing, that in the presence of two inhibitor, your divergence is lower than in the presence of one inhibitor. So basically, what it seems to be suggesting that if you can somehow prevent R5 in your network, you can get some kind of a partial rescue in your system. And turns out that there is some experimental report from the 2019 which kind of supports that idea, that in some situation the microbes can indeed leverage this kind of mechanism wherever you have this some kind of a circular feedback loop. If you want to block one particular reaction, by diluting down the dependence of the other arm, you can actually get back some degree of homeostasis in your system. So it's kind of interesting that, as you can see, this is a very fairly simple model, but this kind of simple model can give you this kind of interesting insight, which is realized in real experimental system. And obviously, for fun, you can make this thing even more complex. Now, instead of two, if you have three, I1, I2, I3, for example, And these different combinations sort of give you different degrees of perturbation in your system. Okay. So for example, this can be one interesting way to look at, like if your real system has this kind of loop in your system, this different combination of antibiotics will give you different degrees of perturbation. Okay, so this is sort of like a very systematic way to think about how to come up with a novel cocktail therapy, for example. So now looking at the real system, which is the folate pathway, the first question we asked that what is the topology of the network? So in order to quantify the topology, there are several ways. Yeah, 5 minutes. Sure. Okay. In order to quantify the topology, we can do several things. So one of the thing is we can compute something called shortest path length, right? So on the right-hand side, we have a random graph consists of same number of nodes and edges. On the left-hand side, we have the number coming from the actual pathway. And similarly, we can do this parameter called transitivity. So to put this thing together, we realized that there's this, in graph theory, there's this classical idea of small world network. And it turns out that if we can compute this parameter sigma, if sigma is greater than 1, then it is said that your network has some kind of small worldedness. And it turns out that for our folate pathway, the value is around 1.8. So it has some kind of a small world-like network, which means that it has this local loops like this triplet-like structure, but those triplets are kind of far from one another. So this is like another way to regulate your network such that if you get some kind of attack from outside, your network has enough resilience to nullify that stress, basically. So basically, as I mentioned, we have several reactions. We made some assumptions about the reaction parameter, and the first parameter we compute is something called a stability index. It's a very coarse-grained metric to sort of figure out that what fraction of the species sort of converges to steady state versus the number of species that is divergent. So the stability index would simply mean that the number of species converged over total number of species in your system. So we can do this titration experiment. Let's say we have this influx and outflux to the network, right? So we can titrate out these two numbers with some given values of Km and Vmax. And you can see that the system, this pathway has some kind of a capacity, because after, if you ramp up your influx rate beyond certain point, you start to get your system start to get less stable and stable. So and this happens because of this interplay between influx and Vmax, because Vmax is the sort of the upper limit of chemical processing capability. So this is sort of the summary, and it's got a quite a busy plot. But the point here is that there are different combination one can think of, which will create different degrees of perturbation to your system. And by carefully analyzing this kind of computational experiment, one can eventually try to think of some combination therapy which will be like most successful in perturbing the physiology. And one of the follow-up of that kind of thinking would be, let's say we have some perturb profile by inhibiting certain reaction. Now one can actually simulate this system by doing some kind of evolutionary algorithm to see that what kind of escape strategy is easy or difficult to escape. So this is now fully in silico, but so we can explore, in fact, different degrees of freedom. And once we have some understanding, then we can embed space into the picture and ask questions like, just by graphical topology, we have some insight, but does that insight change if we include something like space into our framework, right? So those are the kind of things that we want to do next with it. And with it, I thank my mentor and the members in the group, and I'd be happy to take any question. Thank you.
Questions from someone under 30? Oh, 25? Hello? All right, I'll ask a question. In the rescue that you showed, where you're inhibiting both sides of that loop, can you give me some intuition for the reason for that rescue? Is there something I didn't quite follow that?
Yeah, it's a fairly non-trivial actually. I think it has something to do with the nonlinearity of the system, and so nonlinearity and the reversibility of the reaction. So basically, the way it's cycling all the material, the depending on the location of the checkpoint, things can move up and down in a fairly non-trivial way. Yeah.
Okay.
So in other words, it won't be possible if it was just a linear pathway, for example.
So the loop is required then.
Yeah. It's happening purely because of the loop.
Okay.
And this idea of small world network we mentioned, I think it probably points towards the fact that in most of this metabolic network we have this tiny little loop, and I think it's one of the regulatory mechanism to take care of this kind of quote unquote buffering aspect of it to nullify some of the stress maybe.
Oh, interesting. Okay.
Yeah.
You do have a question.
So thanks for your talk. So you gave that toy example that the enzyme will be flux limiting, basically. We have done some work on this and some other people as well. So the co-substrates, things like ATP, NADH, etc., their turnover can be flux limiting as well in a linear pathway.
Yeah.
And so I'm curious about this because the folate cycle has several NADH-linked reactions. So do you model NADH dynamics in your model?
So far, the current version is, let's say, A plus B goes to C plus D. We are trying to figure out, we are trying to represent as A goes to B sort of a representation, but eventually we want to include the cofactor and other type of dynamics as well. So far we ignore, for example, metabolic water and those kind of things. They also might play some role at some points, but I think we started from very simple and we want to build from there. Yeah.
Anybody else? All right, let's thank the speaker one more time.
Thank you.